So this is your penultimate patho topic, and this one's going to be all about the liver. In this first part, we'll look at the underpinning. So we're going to look at the pathogenesis of Hep C, but also of alcoholic liver disease. That then combines really after the initial pathogenesis into the progression through from steatohepatitis to fibrosis and ultimately to cirrhosis if left untreated or corrected. In the second part, we'll look at the applied pathophysiology, so we'll look at signs and symptoms. So I know we covered some of this in a handout beforehand, but just to go over it again, we need to look at this major circulatory flow in terms of the abdominal organs. So the gastrointestinal and hepatic vasculature is sometimes called the splanchnic circulation. And within each of those organs you see up the middle there, we have these splanchnic beds, these capillary beds that form part of this flow. On the right you can see we have our arterial flow. This constitutes a quarter of all of the cardiac output. So this goes through all of the GI organs, but also we have a direct feed of arterial blood via the hepatic artery to the liver. Once the blood has been through the abdominal organs, the other abdominal organs, it then feeds into the portal veins, the hepatic portal system. This also feeds into the liver. Both of these circulations feed into those sinusoids in the lobules. So the splanchnic beds are these microcirculations, these little capillary beds within the tissues of the organs. The preportal veins and portal vein form the hepatic portal system, and they, alongside the hepatic artery for arterial blood, feed the sinusoids. Everything then drains via the hepatic veins into the inferior vena cava, goes back into the heart, and so on and so forth. So a liver lobule then, remember we've got lots of these forming the liver, we have this structure with these vessels running through it. These vessels are called sinusoids. They're still normal blood vessels, they're just running between hepatocytes in this lobule shape. So blood from the portal vein and the hepatic artery flows into the sinusoids between the hepatocytes in each lobule. The blood then flows from the sinusoids into the central vein of the lobule, and then eventually it will get to the hepatic veins, and from the hepatic veins it will return to the heart via the inferior vena cava. So a quick look at a sinusoid from above, if you like. We can see here on the right we have our central vein, so our blood flow is coming in from the left. So we have the portal vein there and the hepatic artery both flowing into the sinusoid. We also have some bile, but that runs outside of the sinusoids, also between hepatocytes. Either side of the sinusoid, this is a normal blood vessel, so we still have endothelial cells, those simple squamous endothelial cells lining this specialist blood vessel. They have little windows in that, if you remember way back, we talked about fenestrations. We will talk more about those later. Within the sinusoids, we also have Kupffer cells. These are our tissue macrophages of the liver. Between the endothelial cells and the hepatocytes being the orangey blobs there, we have a little space called the space of Dis. Within that space, we have something called hepatic stellate cells. Now, generally, they're not that important. They store vitamin A. But actually, in terms of liver pathology, they become very important. So we will come back to those. So we have our venous blood flowing in via the portal vein. We have arterial blood flowing in from the hepatic artery. That then flows out to the hepatic veins and the inferior vena cava and back to the heart. And there you can see we have our hepatocytes, which of course are epithelial cells, but we also have the other epithelial cells involved here, the endothelial cells, the ones lining the sinusoids. So again, some of this was covered in perhaps even more detail in the handout on anatomy and physiology of the liver. But just to summarise it, and just in case you didn't read that, if you didn't read it, please do read it immediately after this video. But we can summarise the functions of the liver in terms of metabolic functions, and this is about the breakdown or detoxification of things like toxins, hormones and drugs. We also have synthesis. So the liver is responsible for synthesizing bile. It synthesizes amino acids in that it's able to transfer amino acids from one base to another. So it can synthesize a different amino acid from an existing one. Also the synthesis of various proteins, various chemicals that go throughout the body. Carbohydrate and lipids are also drawn together, metabolized and synthesized by the liver. In terms of lipids, of course, we're talking about very low density lipoprotein being released from the liver, and those are little packages of lipids. We have storage functions. And we know about glycogen. If you remember, glucagon from our alpha cells, our pancreatic alpha cells, stored excess glucose as glycogen for release when we need it to convert it back to glucose. But we also have the storage of lots of vitamins, and I mentioned stellate cells. Stellate cells store vitamin A. A lot of the vitamins, the extra stores of vitamins, are stored within the liver. If you remember, our B vitamins are stored here. And various minerals like zinc and copper and things like that. Finally, we do have an immunological function, because if we have blood coming, venous blood coming from the gastrointestinal tract, if pathogens have managed to evade the GI tract defense mechanisms and get into the hepatic portal system, the liver is the next line of defense. So it provides this sort of final defense before the blood flows back to the heart against any ingested pathogens. A lot of this defense, of course, is supplied by Kupffer cells, those tissue macrophages that sit within the sinusoids. So that was a little reminder about the anatomy and physiology of the liver. So let's start to talk about what goes wrong. In terms of liver disease, the underpinning pathophysiology we're going to look at are the pathogenesis in terms of Hep C, hepatitis C virus, and also alcoholic liver disease. Of course, other diseases are available, but these are the ones we're going to talk about because these are really the most common. And in terms of these, once they've been stimulated, we need to look at the progression, because in this situation we have a chronic inflammatory state. So we have this mixed morphology of damage and repair. This starts with a fatty liver known as steatosis. And we'll talk about those are two different mechanisms in terms of Hep C or ALD. Then that moves into a mix of fat and inflammation. That fatty liver stimulates an inflammatory response, of course. So we end up with steatohepatitis. Ultimately, if the cause is not removed, we will now start to go into a really chronic inflammatory situation that results in fibrosis and eventually cirrhosis. Another cause, this is one of our other ones that are available, of liver diseases is this non-alcoholic steatohepatitis. There's various abbreviations. It could be NASH, it could be NAFLD. I think NASH is about the most common. And this is where liver disease is associated with a mix of obesity, diabetes, hyperlipidemia and hypertension. In other words, this whole metabolic pathology. So first of all, let's have a look at hepatitis C virus. Another little virus. So what it is, this is what we need to know, isn't it, in terms of replication? It's an enveloped single sense strand RNA virus. And of course, we have a high error rate during RNA virus replication. Remember, in a single sense strand, what it's doing, it has to make its viral proteins by using its single sense strand in our own ribosomes to make the viral proteins. It then has to make an antisense strand to use as a template to make more sense strands for the virions. And it's these double-strand RNA interactions that cause quite a high error rate in terms of replicating that RNA. So we end up with many mutated variants. This gives us a bit of an issue because finding an effective vaccine is then very difficult, because we find a vaccine that will recognize a particular protein on the virus, that virus mutates, the protein changes, our vaccine is no longer effective. Transmission is by blood-to-blood contact. And in the developed world, really, most infections are due to the use of injected drugs. Once it gets into the blood, Hep C often binds to LDL or VLDL. And it's this affinity for the lipoproteins is thought to be why HCV generally binds only to hepatocytes. So our LDL becomes bound with our Hep C, so Hep C goes along, binds to LDL. The LDL then binds to LDL receptors on hepatocytes, and our cheeky little Hep C virus then sneaks its way in alongside the LDL. And if you remember, LDL is endocytosed via this receptor-mediated endocytosis, which involves the formation of these clathrin-coated pits. So here's our LDL sitting there all happily about to go and bind to its LDL receptor on our hepatocyte. Whack! In comes HCV, binds to the LDL. LDL, yeah, well, I'll still go along and bind to my receptor. So it binds to its receptor, and both of them are then endocytosed via the clathrin-coated pit. Then, of course, everything's going to be broken down by a lysosome, receptors recycled, LDL will be used, but HCV is already in the cell. Once it gets into the cell, of course, like other viruses, other coated viruses, it needs to uncoat. So it uncoats, its sense strand is now ready and free and goes and binds to a host ribosome to be read. This then produces viral proteins. These are the proteins that are going to make the capsid, make the RNA-dependent RNA polymerase, those sort of proteins that viruses need other than the nucleic acid. We need the nucleic acid though, of course, because what we need more copy sense strands for the virions. So RNA-dependent RNA polymerase makes a template to then form these copy sense strands. The virions are assembled, they bud from the hepatocyte, and of course once they're free they can go off and infect other hepatocytes. So what are we going to do about this? Well, we've got our Kupffer cells. These are our tissue macrophages within the sinusoids. They're able to phagocytose the infected hepatocytes, but also free virions. The activated Kupffer cells secrete cytokines, prostaglandins, chemokines, colony stimulating factors when they phagocytose. And of course, this is going to upregulate the whole of the response. So these mediators initiate leukocytosis of first neutrophils, then additional monocytes, which will become macrophages, natural killer cells, and dendritic cells. They also facilitate the vasoactive changes, cell adhesion molecule expression, and therefore emigration and eventually chemotaxis. And from that you can see we've got all of the phases of a normal innate response being stimulated. We then have the potential to create antigen presenting cells once these phagocytes are phagocytosed that can then go off and activate the adaptive response against, specifically, HCV. In any viral infection, the innate mechanisms that we've talked about before that are most relevant are the interferons and natural killer cells. Interferons, if you remember, are secreted by infected cells, virally infected cells. The interferons then bind to adjacent cells. They use a second messenger to stimulate the infected cell to synthesize antiviral proteins. The antiviral proteins degrade intracellular viral RNA and stop double-strand interactions. In other words, they're going to stop the hepatitis C virus from replicating. So it inhibits viral replication. But of course it often causes apoptosis of the host cell. And it does this through the upregulation of p53. Remember, this is our gatekeeper tumour suppressor gene. And its role, if the cell is beyond repair, is to trigger apoptosis. Natural killer cells, these are a really well recognised defence against virally infected and incidentally malignant cells, because these cells don't display MHC1 usually. And if you remember, when we first talked about natural killer cells, we talked about them operating within the innate response, and we talked about them having these two receptors. One binds, the other one is looking for host cell MHC1. If they don't find the host cell MHC1, they then release perforins and granzymes to kill that cell. They can also become antigen specific and operate within the adaptive response if antibodies bind to them. If you remember, the antibody that has FV regions that are antigen specific, the antibody can bind to a natural killer cell via the FC and then bind to the target. And this will also activate the natural killer cell to release perforins and granzymes and kill that target cell. So the natural killer cell releases perforins and granzymes, they activate the death pathway in the target cell and cause apoptosis. In addition, we have activation of the adaptive immune response. So Kupffer cells, recruited macrophages, dendritic cells can all become antigen-presenting cells. They travel to the hepatic lymph nodes, which sit just above the liver, and they present various viral antigens to activate naive T cells and B cells. Of course, what we then get, we get activated B cells that become plasma cells and secrete antibodies that are specific for the antigens from the hepatitis C. We also get antigen-specific T helper cells and cytotoxic T cells. And they could be specific to multiple epitopes displayed by the hepatitis C virions. Antibodies then agglutinate any virions, any free virions, and opsonize them. They can also assist natural killer cells, as we just talked about, by the process of antibody-dependent cell cytotoxicity. Cytotoxic T cells, similar action to natural killer cells. They can bind to infected cells via viral proteins displayed in an MHC1, and they can release their perforins and proteases to cause virally infected cells, hepatocytes, to go into apoptosis. Okay, so how does this all translate into this steatosis of the liver, fatty liver, and steatohepatitis, where we get the fatty liver and inflammation? Steatosis develops because HCV, if you remember, we had a similar situation with TB. HCV alters intracellular lipid metabolism to support their own replication. It stimulates lipogenesis, so in other words, the formation of triglycerides from acetyl-CoA, reduced lipid secretion from the hepatocyte, and if you remember, hepatocytes release very low density lipoprotein, so lots of fat, not much protein. So we're losing less lipid, we're producing lipid within the cells. And there's also reduced beta oxidation of lipids. So there is a reduction in the breakdown of fatty acids to generate acetyl-CoA. So there are going to be more fatty acids hanging around within the hepatocyte. As a result, we get this collection of lipid droplets and cholesterol developing within the hepatocytes. And this causes what we call fatty liver, steatosis. Steatohepatitis, of course, is going to develop because, as we've already said, we stimulate an immune response. As a result of that, we've got the secretion of all these cytokines, colony-stimulating factors, all the other inflammatory mediators. Plus, we've got apoptosis and necrosis of these infected, fat-laden hepatocytes. This is all going to upregulate inflammation. So, in addition to having steatosis, fatty liver, we've now got inflammation, which gives us steatohepatitis. Hepatitis C can be controlled, it can be resolved, and we can treat it. But in some hepatitis C patients, they develop a weakened immune response due to HCV. And in particular, this affects the efficacy of interferons and natural killer cells, are two of our key mechanisms against viral infection. In addition, what's also been seen is that the level of circulating T helper and cytotoxic T cells can be reduced. So some people have a weakened immune response to Hep C. The exact mechanisms aren't fully understood, but of course, if we can't get rid of hepatitis C, this can then develop into a chronic infection. Chronic hepatitis C will then stimulate the progression through from steatosis and steatohepatitis to fibrosis, cirrhosis, and potentially hepatocellular carcinoma. And around 30%, that's a third, of HCC, hepatocellular carcinoma cases in the Western world are due to chronic hepatitis C infections. It is still relatively rare, but it can also take a lot of years to develop. Which brings us on to our other pathogenesis that we're going to talk about, which is alcoholic liver disease. So alcoholic liver disease is one of the most common causes of liver cirrhosis. But again, it still can take quite a while to develop. It can take 10 years of heavy drinking to develop. This is in some ways dependent upon other factors. So aside from heavy alcohol intake, the determinants of progression are genetic factors, environmental factors such as diet, comorbidities such as obesity, metabolic syndrome, all of those things, other liver disease, including HCV, and medication. So let's talk about how we deal with alcohol. The chemical formula of alcohol, ethanol, is CH3CH2OH, just in case you're interested. Doesn't make it quite so interesting to drink, does it? Most ethanol is oxidized in the liver by two possible pathways, although the primary pathway is the alcohol dehydrogenase ADH pathway. In this pathway, alcohol is converted to acetaldehyde and then into acetic acid. During both phases of this process, protons H+ from ethanol are transferred to NAD+. Remember NAD+? In the electron transport chain, it was our electron carrier. So it also involves the binding of 2 electrons, but it's mopping up the protons, the H+ from ethanol. As a result, the hepatocytes produce NADH. This will eventually, if we increase the amount of alcohol, will create an excess of NADH in the liver. And consequently, because we're using NAD+, there will be a depletion of NAD+. Eventually, when we run out of NAD+ to convert to NADH by mopping up these protons, we activate the next pathway. This is the microsomal ethanol oxidizing system, otherwise known as MEOS. MEOS doesn't rely on NAD+ to convert alcohol. It does increase cytochrome P450, which you may have come across if you've done your non-medical prescribing, which has multiple effects on drug metabolism and biological function. This is a significant cytochrome in the liver involved in detoxification. So alcoholic liver disease, we also have this concept of progression. The first stage is still steatosis. It's found in most excessive drinkers, but this is reversible with a reduction in alcohol intake, obviously. Eventually, this may develop into steatohepatitis in up to 1/3 of people who continue to drink heavily. And here, of course, we've not only got that fatty liver, we've also got inflammation. Potentially, that could lead to liver failure. In the 3rd stage, fibrosis and cirrhosis develops, but only in about 10% of heavy drinkers. And as we said, it can take a long time to develop. And it is more likely if there are other hepatic disorders. Cirrhosis, we'll talk in detail a little bit about what happens in terms of cirrhosis later. It's a diffuse fibrosis of the liver. And what happens is the normal liver architecture is converted into these structurally abnormal fibrotic nodules. Hence the nodular feel in cirrhosis when we palpate the liver. So let's have a look at ALD in terms of this development of steatosis and eventually steatohepatitis. In people who drink more than 60 grams of alcohol per day, I had to do a little looking up and calculation to work out what that was in units. It's about 7.5 units. Hepatocyte lipid metabolism becomes altered due to many different sort of complex mechanisms, one of which is that NAD+ depletion that we talked about. As a result, the hepatocytes accumulate fat intracellularly, just like with hepatitis C. So, like in hepatitis C infection, it stimulates lipogenesis, the formation of triglycerides, if you remember from acetyl-CoA, reduced VLDL secretion from the hepatocyte, so the hepatocytes hold on to lots of fats, and reduce beta oxidation of lipids. So the hepatocytes become full of fatty acids that aren't used to generate acetyl-CoA. This lipid accumulation causes oxidative stress and apoptosis via intrinsic death pathways. And this then leads us in really into steatohepatitis, because of course the apoptosis will trigger an inflammatory response. When we get an inflammatory response, we get steatohepatitis. We also might have a situation where we get altered protein and lipids being released from these cells, and this may stimulate an adaptive response, although there's not an awful lot of research on the adaptive response in alcoholic liver disease. So both of our conditions that we've talked about in terms of pathogenesis, we've taken them through steatosis to steatohepatitis. Both of these can progress in exactly the same way onto the other stages of fibrosis and cirrhosis. So our next stage after steatohepatitis is fibrosis. And this is due to the activation of those innocuous little stellate cells. Hepatic stellate cells, sometimes called HSCs, are activated, and when they are, they cause the deposition of collagen, which gives us fibrosis. Those stellate cells, if you remember, we find in the space of Dis, so between the endothelium and the hepatocytes. And usually they're very helpful. They store vitamin A in lipid droplets. But in an inflammatory situation, they can become activated by various inflammatory mediators. One is platelet-derived growth factor, PDGF, and two of our big boys, TNF-alpha and interleukin-1. Also, they can be activated by transforming growth factor beta, TGF-B. This is a cytokine that's secreted by all leukocytes, and its secretion is induced not only by inflammation, but also by acetaldehyde. Remember, that's what alcohol was converted to in our primary metabolic pathway of alcohol. Ironically, apart from activating hepatic stellate cells, TGF-beta is secreted from hepatic stellate cells. So it's upregulating itself. Now, our TGF-beta, transforming growth factor beta, does other things as well. It increases collagen synthesis. It increases TNF-alpha secretion, and it inhibits matrix metalloproteases, MMPs. So, in other words, what it's doing, it's increasing the amount of collagen being synthesized. It's also increasing the amount of TNF-alpha, which helps to activate HSCs. And it's also inhibiting the things, the proteases, the MMPs, that would normally break down that collagen. So let's just have a look at normal. In normal sinusoids, we have our Kupffer cells within the sinusoids, and then in the space of Dis, we have our hepatic stellate cells, sitting there nicely storing vitamin A for us. So what do we get when we activate HSCs? we get HSC proliferation and migration. So we get an increase in numbers, and they also start to move. They start to move into the sinusoids. They start to move into the liver parenchyma, away from the space of Dis. The HSCs go through metaplasia. They change from being HSCs and they become more like myofibroblasts. Fibroblasts, of course, secrete collagen. Myofibroblasts secrete a very elastic form of collagen. They're the ones that we talked about way back as being responsible for pulling wound edges together so to facilitate repair, so there's less of an area to repair after tissue damage. And we get a generation of the large amounts of collagen because of this, and other extracellular matrix components. And this, of course, is stimulated by TGF-beta secretion from the transformed hepatic stellate cells. And remember, TGF-beta is also stimulated and increased from all leukocytes by inflammatory mediators and by increases in acetaldehyde. So the more alcohol we drink, the more acetaldehyde we'll have, and the more we will stimulate TGF-beta. And apart from inducing collagen synthesis, and don't forget TNF-alpha secretion, which gives us more hepatic stellate cells, TGF-beta inhibits proteases that would normally be breaking down collagen. So we have fewer proteases to break down the collagen that's being produced. In addition, we have an increase in the secretion of tissue inhibitors of metalloproteases, known as TIMPs. Now, TIMPs normally work with MMPs in a very fine balance to try and control the action of MMPs so they don't break down too much collagen. So TIMPs inhibit the breakdown of collagen by matrix metalloproteases. So if they're increased, we've got even more inhibition of MMPs. So collagen is not broken down. So let's have a look at our hepatic stellate cells again. There we are where we left them, looking all innocuous. They go through metaplasia when activated. So they transform into myofibroblasts. They then proliferate and migrate. And then they secrete collagen. So primarily stimulated by transforming growth factor beta, they start secreting lots and lots of collagen. So how does fibrosis and a few other changes affect how the liver functions? Well, for a start off, those endothelial cells lining the sinusoids, remember we said have fenestrations. They have little windows that pass all the way through them, through their cytoplasm. They also have an incomplete basement membrane. Of course, this is going to make that endothelial lining of the sinusoids very, very permeable, which, if we're bringing in nutrients for the liver to deal with, it is very helpful. So it allows the absorption of nutrients from the hepatic portal system. Due to growth factors, in this case, there is a closure of the fenestrations and a thickening of the basement membrane, and this is before fibrosis starts. And there is also loss of the microvilli on the hepatocytes. So of course that's going to reduce their ability to absorb any nutrients. So we've put up a bit of a barrier in that we no longer have our fenestrations or our permeable basement membrane, but also we've lost the microvilli on the hepatocytes that would have been able to absorb those nutrients. So we are going to have a bit of a problem in terms of malabsorption. So permeability to the enteric solutes that we want is reduced by the loss of fenestrations and the impermeable basement membrane, and the loss of hepatocyte microvilli. And then fibrosis starts. So fibrosis, or that collagen being laid down in the space of Dis and surrounding the hepatocytes, is going to put up another barrier. That fibrosis also is going to prevent any absorption of the enteric solutes. So the progression on from fibrosis could be cirrhosis. It's the late stage of liver disease. It's a combination of chronic inflammation with its usual concurrent damage due to necrosis and apoptosis of hepatocytes, plus repair and the proliferation of hepatocytes. Remember, these are epithelial cells, so they are highly regenerative. There is also going to be the replacement of functional hepatocytes with fibrosis. Fibrosis that's not going to be broken down by MMPs for the reasons we've already talked about. This fibrosis is going to cause obstruction of the sinusoids and of the space of Dis, and ultimately of the portal vein. And as we just talked about, there will be malabsorption of enteric solutes because of the loss of those fenestrations, the changes in the basement membrane, the fibrosis, and the loss of microvilli on the hepatocytes. At this stage, steatosis is reduced because it's now more about this progressive fibrosis and the necrosis of the hepatocytes. And as we said, we get quite high levels of hepatocyte proliferation in an attempt to replace the necrotic or apoptotic hepatocytes. This benign hepatocyte proliferation forms nodules. These are prone, because of this high proliferation of course, to develop into malignant cells, to develop into hepatocellular carcinoma. And just to really emphasize these nodules, we have these bands of fibrous tissue known as septa, and they separate out these proliferative hepatocyte nodules, and this enhances that nodular look and nodular feel. So here we can see some actual histology, and the blue that we can see there are the fibrous bands. This is our fibrosis, our septa, separating out the nodules of regenerating hepatocytes. Okay, so that was our underpinning pathophysiology for hepatitis C and alcoholic liver disease. Our next section will look at the applied pathophysiology of signs and symptoms in liver disease generally. But before you move on, make sure you've understood this. do all your usual, making your own notes and diagrams. Ideally, discuss it with a study buddy so that you know you've got it right, and also to prompt your recall. Don't delay, because I'm on final countdown here now, you can tell. The exam is only 7 weeks away.